Welcome to this comprehensive exploration of functional brain imaging using electromagnetic neurotechnologies. In this video, we will cover a variety of techniques employed in understanding the human brain's activity, ranging from electroencephalography, EEG, to more advanced methods like positron emission tomography, PET, and emission computed tomography, SPECT. Each technique provides unique insights into brain function, offering unparalleled opportunities for both clinical and research applications. Stay with us as we break down the different modalities, their data acquisition, and processing methodologies. In this slide, we see how various brain imaging techniques relate to the electromagnetic spectrum. These techniques utilize different parts of the spectrum to capture brain activity in unique ways. SPECT and PET operate in the gamma ray region, capturing metabolic activity in the brain through radioactive tracers, helping to diagnose stroke and Alzheimer's. fNIRS uses infrared light to measure blood oxygenation changes in the brain, offering a portable way to monitor brain function during real-world activities. fMRI works in the radiofrequency and magnetic regions, measuring blood flow and oxygenation to provide detailed images of brain activity. MEG detects magnetic fields from neural activity, using the low-frequency range to capture real-time brain dynamics with high precision. EEG measures electrical activity directly from the scalp, providing insights into brainwave patterns, useful for diagnosing epilepsy and studying sleep. Each of these technologies offers into brain function, contributing to a more comprehensive understanding of the brain's activity. In this slide, we examine the processes involved in EEG and MEG data acquisition and processing. These two neuroimaging techniques are both powerful tools used to study brain activity, though they capture different aspects, electrical versus magnetic signals. We begin with EEG. Electroencephalography measures the electrical activity of the brain by placing multiple electrodes on the scalp. The first step in EEG data acquisition is recording brain activity from the surface of the scalp, where the electrical signals from neurons are captured. These signals, or brainwaves, are then converted into time series data. The next, the signal by filtering out noise and artifacts, preparing the data for further analysis. This stage also includes digitizing the data using analog-to-digital converters, A/D. The processed EEG data is then subjected to advanced techniques like blind source separation, pattern recognition, and topography to extract meaningful features from the raw signals. These methods help identify patterns related to cognitive states, such as attention, relaxation, or sleep. On the other hand, MEG, magnetoencephalography, measures the magnetic fields generated by the electrical activity of neurons. The first step in MEG data acquisition is recording the magnetic signals from the brain using highly sensitive SQUID, superconducting quantum interference devices, sensors placed around the scalp. Sensors detect the faint magnetic fields produced by neuronal currents. Like EEG, the recorded data is processed by filtering to reduce noise. Then, the beamformer projection technique is applied, which maps the extracranial magnetic data back into the brain space to visualize the areas of the brain producing activity. The next step involves estimating the phase and amplitude of brain waveforms using the Hilbert transform. These parameters are then analyzed to study phase synchronization, amplitude cross-correlation, and other temporal dynamics, which are critical for understanding brain network activity. Finally, an adjacency matrix is a map of functional connectivity between brain regions, offering insights into how different regions interact during various tasks. Both EEG and MEG offer real-time insights into brain dynamics, but they complement each other by measuring different types of signals, electrical activity with EEG and magnetic fields with MEG, leading to our understanding of brain function from complementary perspectives. Now, we shift to magnetic resonance imaging, or MRI, which provides detailed structural images of the brain. MRI is based on the principles of magnetic fields and radio waves, offering highly detailed 3D images of brain structures without the need for any radiation. The data acquisition process involves placing a magnetic field and using radiofrequency pulses to excite hydrogen atoms in the brain. When the hydrogen atoms return to their original state, they emit signals that are captured to construct a detailed image of the brain's anatomy. MRI is invaluable for diagnosing brain conditions like tumors, strokes, and multiple sclerosis. In addition, in this slide, we see the step-by-step process of how MRI captures brain activity. First, a stimulus, like a visual cue or cognitive task, triggers a neural response, shown in the top part of the slide. This activates brain areas, increasing metabolic activity and blood flow. It illustrates how synaptic signaling and metabolic signaling lead to a vascular response, where blood vessels dilate to deliver oxygenated blood to active brain regions. The bold signal relies on changes in oxygen levels in the blood. As shown, deoxygenated blood causes MRI signal dephasing, and oxygenated MRI signal, highlighting active areas. The MRI scanner, shown below, captures these signals as case-based data, which is then processed using an inverse Fourier transform to form the final brain image. This process allows us to see which brain regions are active during tasks, with stronger signals indicating more oxygen. Through this process, MRI gives us both structural and functional insights into the brain's activity, enabling detailed imaging of brain functions in response to various stimuli. In this slide, we are exploring functional near-infrared spectroscopy, or fNIRS, which is a non-invasive optical technique used to measure brain activity. The first image illustrates how infrared light is used to detect changes in blood oxygenation levels in the brain. When neurons become active, they require more oxygen, increase in blood flow to the active regions, which can be detected by fNIRS. The technique works by shining near-infrared light through the skull, and the changes in light absorption caused by the varying levels of oxygenated HBO and deoxygenated hemoglobin HBr are recorded. As shown in the graph, these neural activity, allowing us to map which regions of the brain are engaged during different tasks. fNIRS has several advantages, including its portability, safety, and relatively low cost, which makes it ideal for use outside the lab. The image in the bottom left shows a typical fNIRS setup, where the sensors are to monitor brain activity. This setup is especially valuable in cognitive neuroscience research and is used in clinical environments, such as during rehabilitation, where real-time brain function can be monitored. The DOT, diffuse optical tomography, image reconstruction shown in the bottom center depicts the process of mapping the data collected from multiple sensors. This method involves high-density imaging arrays, with measurements taken at different distances, to improve the resolution of the brain activity map. Forward light modeling and sensitivity profiles are used to refine these images, helping us better understand how brain regions respond to cognition. Overall, fNIRS provides a powerful, non-invasive method for studying brain function, both in the lab and in real-world environments. It is increasingly used in a variety of settings to monitor brain activity in individuals during different tasks or conditions. In this slide, we delve into positron emission tomography, or PET, a technique used to measure brain metabolism. The process begins with labeling, where a chemical molecule is combined with a positron-emitting isotope to create a PET tracer. This tracer is designed to bind to specific molecules or active areas in the body in the brain. The labeled tracer is then injected into the bloodstream, where it circulates and accumulates in regions with high metabolic activity, such as active brain areas. As the tracer accumulates, it begins to emit positrons, which collide with electrons in the body, leading to annihilation. This is the release of gamma rays, which are detected by the PET camera. The detectors in the camera capture these gamma rays in two opposite directions, which allows us to pinpoint the exact location of the metabolic activity. The signals are processed from multiple channels and combined to create a detailed image of brain activity. The data processing unit, where it undergoes image reconstruction. The reconstructed PET image shows areas of the brain with high or low metabolic activity. This helps identify regions that may be overactive or underactive, which is particularly useful in diagnosing conditions like Alzheimer's, Parkinson's disease, and brain tumor. PET provides valuable insights into both the structure and function of the brain, offering a unique look at brain activity in real time. In summary, PET is an invaluable tool for understanding brain function and diagnosing neurological diseases, providing essential information about brain metabolism and blood flow. In this slide, we focus on single-photon emission computed tomography, SPECT, a technique that is similar to PET but uses a different approach. SPECT is used to measure blood flow in the brain rather than metabolic activity. The process begins when a radioactive tracer is injected into the bloodstream, as shown in the diagram. The tracer emits gamma rays that travel through the body, and these rays are detected by a rotating gamma camera. The camera captures the gamma rays from various angles, providing the data needed to create detailed 3D images of brain activity. The SPECT detector captures the emitted gamma photons and the system's electron signals. The image on the right shows how the scintillation photons are captured and converted into signals by the detector's light guide and photomultiplier tubes, PMTs. The energy discriminator then helps refine the data, distinguishing relevant signals from noise, and the display monitor shows the final image of the gamma emitter distribution. SPECT is particularly useful in clinical settings to diagnose conditions like epilepsy, stroke, and dementia. It helps doctors visualize abnormal blood flow in specific regions of the brain, which is crucial for diagnosis and treatment planning. By providing valuable information about the brain's vascular health, an essential tool in both diagnosing and monitoring neurological diseases. In this slide, we examine the reconstructed images of various brain imaging modalities and how each technique processes raw data into valuable brain images. The comparison table at the bottom offers a detailed look at key characteristics of each method, providing insights into their spatial resolution, temporal resolution, and the activity they measure. On the left side of the slide, we see images from SPECT and PET. These techniques measure metabolic activity in the brain. The raw data from gamma ray emitters is reconstructed into images, as seen in the PET image, which highlights metabolic brain activity. SPECT similarly focuses more on blood flow. PET has a higher spatial resolution, 4 millimeters, compared to SPECT, 2 to 3 centimeters, and it measures metabolic activity, unlike SPECT, which primarily measures blood flow. Moving to the fNIRS image, we see a technique that uses near-infrared light to measure change in concentration, related to changes in blood oxygenation during brain activity. The spatial resolution of fNIRS ranges between 10 to 30 millimeters, and its temporal resolution is about 20 milliseconds, making it suitable for measuring hemodynamic activity in real time. This technique is particularly valuable and non-invasive nature. fMRI and MEG images, which are at the center, show high-resolution brain images and capture dynamic brain activity with impressive precision. fMRI has excellent spatial resolution, 2 to 3 millimeters, and measures hemodynamic activity, changes in blood flow related to neural activity. It has lower temporal resolution of 1 second compared to MEG, which captures the magnetic signal generated by neuronal activity with a spatial resolution of less than 1 centimeter and a temporal resolution of less than 1 millisecond. MEG is ideal for studying real-time brain dynamics, like sensory processing, with its high temporal precision. We have EEG, which provides the electrical signal directly from the brain. EEG has the highest temporal resolution of less than 1 millisecond, making it invaluable for studying fast brain processes like seizures or real-time cognition. However, its spatial resolution is lower, at 2 to 3 centimeters. It covers cortical brain regions, as shown in the image. In summary, each of these modalities has unique strengths and trade-offs. EEG and MEG provide unmatched temporal resolution, allowing for real-time brain activity mapping, but they have limited spatial resolution compared to fMRI and PET. Offers high spatial resolution and is ideal for studying brain structure and function in detail, but its temporal resolution is slower. fNIRS provides a balance with good portability and moderate spatial and temporal resolution, while PET and SPECT excel at measuring brain metabolism and blood flow, making them useful for clinical applications diagnosing Alzheimer's or tracking cancer activity. To conclude, functional brain imaging technologies, especially those involving electromagnetic neurotechnologies, have revolutionized our understanding of the brain. From mapping real-time brain activity with EEG and MEG to visualizing brain metabolism with PET and SPECT, each technique offers a unique lens into the brain's. As the technology behind these imaging techniques continues to advance, we can expect even more powerful tools for diagnosing and treating neurological conditions, as well as furthering our understanding of how the brain processes information. Thank you for watching, and stay tuned for more on the future of brain imaging and neurotechnologies.